recording. And I'll get started with the talk. So I call this talk Experimental Design Matters, like matters related to experimental design, but also it matters how you design your experiments, because a properly designed experiment answers the questions that we care about as scientists, and an improperly designed experiment doesn't. So, you know, it's clearly something important to think about. What are the main points that I'm going to try to convey in this talk? Basically, as scientists, we create models of reality. You know, you all treat a mouse as a model of a human being, right? It's not a perfect one-to-one correspondence, but it's like a model. It represents some important aspects of human biology. But just like we use, you know, statistical models and computer models, simulations, those are like a abstracted representation of reality. A map is a model of reality. Well, an experiment is also a model of the real world. We wouldn't bother doing an experiment if we didn't think it had some bearing on the real world. And so we create these artificial worlds in the lab or in controlled conditions to help us understand what's going on in the real world. By the way, you know, feel free to interrupt at any point, either on Teams or in person, if you have a question or a comment. And like I said, experiments answer scientific questions, and you start with a question, and then the experimental design follows from there, depending on what question you have. But there are sort of trade-offs. There's always advantages and disadvantages to particular designs, which we'll talk about. And you know, reality is complex, it's noisy. There are other effects happening besides what the ones that we're scientifically interested in. But if we properly design our experiment, we can pull out that signal that we're interested in from the background noise. And the better you design an experiment, the better able you will be to do that. So let's go to, you know, this may be review for some people, but honestly, I never really got this fundamentals in my schooling. So I think it is important to talk about. You know, maybe you got it in fourth grade. We talk about the scientific method, but then they never go over it again. So if you weren't paying attention in fourth grade, you missed it. So let's go over it again. What is an experiment? So, you know, you all have heard, these are like really outdated memes at this point, but I still put it up there anyway, just to like illustrate, you know, you have your kind of like very oversimplified idea of the scientific method. An experiment is part of that, where, you know, you make an observation about reality, formulate a hypothesis based on that, and then come up with an experiment to test the hypothesis. So we can define an experiment as a controlled procedure to compare or test a hypothesis. We'll talk about control later on. So we do experiments to gather evidence in favor of or against different hypotheses. And what is important about experiments is that the control enables us to say something about causality. You know, it is not easy from observational data alone to say why something happened, because we don't know what different influences are acting on things in the real world. If in an experiment, we can control everything, set everything equal, except for the one influence that we care about, and then we observe a change in the result, we can say, well, that change was caused by the thing that we are testing, manipulating in our experiment. So yeah, like I said, the scientific method is obviously way more complicated in reality, but that's how experiments fit into that. And as I already mentioned, an experiment is just one of many models we can make of reality. And so I also mentioned there are trade-offs when it comes to scientific studies. Well, the key trade-off when it comes to experiments is control versus realism. So if you go out there and observe and take measurements on people or animals or plants out there in the world, out there in nature, that's very realistic. That's real world data, but you don't have any control over, you know, what those people are eating, what they're doing, what they're being exposed to. So you have very realistic data, but you don't really know why you're getting the data you're getting because you don't have control. And so an experiment, and so obviously the sort of most controlled conditions would be a computer or a mathematical simulation where you can just change parameters in an equation and get the results out. But that's very far from reality, obviously, because it's just a simulation. So an experiment is somewhere in between. And there's sort of a hybrid between experiments and observations, like which some people call natural experiments, and that would be something that you could, if something happens in the real world that is almost like you can act like it's a randomly controlled experiment, even though it's not. Like if you had baseline data on a population and then something happens, like if you have baseline data on a forest and then a wildfire occurs, you can look at before and after the wildfire and measure things and say, the wildfire caused these changes, for example. So that's like a natural experiment. Or you can look at the places that were burned and the places that were not, if you can sort of assume it's more or less random where the wildfire hit. Or, if it's a human population, if a chemical spill occurs, you can say, these people were exposed and these were not. But that's not a true experiment. You still don't know for sure why those changes are occurring, since you didn't fully control those conditions. And an experiment is a model, and the famous quote from a famous statistician is that all models are wrong, but some are useful. Like every model that we do, whether it's a simulation or an experiment in the lab, is not a perfect representation of reality, so it's wrong in some sense, but some models are useful. We can learn something about reality from them, even though they're not perfect. We can still learn from them. That's what he meant by that. And so, like I said, an experiment represents somewhere along the trade-off between control and how close it is to real world. So this, maybe you all won't think it's relevant. I think it's very cool, put the work we're doing into the context of the history of science, at least the sort of Western European paradigm of science and how it developed over time. Basically, human beings experiment. We are an experimental species. Babies, toddlers will push something and see what happens, and they'll manipulate their environment and get data on the effect of that, and that's how they learn about the world. All humans have experimented since the beginning of prehistory, but the sort of first formal, controlled scientific experiment that we can trace back to and still have knowledge of was about 1,000 years ago in the Middle East, where a scientist, the term scientist didn't exist yet, but a very smart dude named Ibn al-Haytham. Back then, people thought that you could see because light, some kind of ray went out of your eyes to the thing you're looking at. And he was able to, through a series of basically experiments, controlled conditions with lenses and mirrors, get evidence saying, well, really, it's actually something going into your eyes, not out of your eyes, that makes you see. So an important piece of knowledge was gained there. And, many people have heard about Galileo about 400 or 500 years ago, who designed experiments, dropping balls from towers and rolling them down ramps. And he tested hypotheses about physics, how forces act on objects. And of course, as you all are sort of in the clinical field, so you may have heard about this famous story of the first randomized clinical trial in the 1700s. There was, the British Navy had a lot of issues with scurvy. If men were on a boat for too long, their teeth started to fall out, and they got very sick and weak and everything. And that was an issue, so they wanted to figure out a cause for this disease and then figure out how to alleviate it. So there were all these different sort of competing ideas about how you could treat scurvy. Well, this doctor named James Lind took 12 sailors and divided them up into groups of 2, and each group got a different treatment for scurvy. So the 2 of the men were given cider, 2 of the men were given sulfuric acid. Hopefully that was, I don't know how much they were given, but some had to drink seawater and things like that. And it was sort of anecdotally known that eating oranges and lemons would cure scurvy. And so, at least according to the story, the sailors that ate the oranges and lemons got better from scurvy, because scurvy is nothing but a vitamin C deficiency. So if you eat vitamin C, the symptoms will go away. So yeah, basically that was a controlled experiment, just like we do now. It was random which men were given which treatment. So then we can say, if we see an effect of the treatment, we can say, because it was random who got the treatment, we can say that that's why they got better. Of course, now we would use more replication, but we'll go into that stuff later. So yeah, then that was kind of a precursor. It took a while for that clinical trial approach to really catch on after that, but it was sort of around the late 1800s, the sort of formal, systematic way of doing experiments that we do now was officially kind of set in writing. And Charles Peirce did an early experiment, which we would now call a repeated measures design, testing how sensitive people's fingers were to pressure. And so that was a key early experiment. And then in the 1920s, a statistician, he was sort of one of the early people who coined the term statistician, R.A. Fisher, did a lot of work on experimental design. And this was in agricultural science, dividing up a field into plots and planting potatoes under different treatments and seeing how well they grew. And so that's why, because a lot of early experimental design work was done in agriculture, people say plots and blocks, even if you're not in the agricultural field. And we'll talk about Fisher more later. And then, as the 20th century went on, experiments, even though a randomized controlled trial was done in the 1700s, the first kind of modern style trial like we would do today was done in the 1940s, testing antibiotics to treat tuberculosis. So, before that, pretty much all medical journals just kind of had case reports. A doctor would have an interesting patient. They would make observations on that patient, write it up in an article, and publish it in a journal. Like, that's really important knowledge, but it can only get you so far. So you need the randomized trials as well. So that was a big milestone. Yeah, and so but before that, pretty much all there was were these anecdotal studies. Although we still see them today. Some diseases are just too rare to do experiments on, and all you could do is anecdotal studies. But before the RCTs, that was all there was. And experimentation also took a big step in industry, especially in Japan after World War II. Japanese automakers and things like that started to do experiments within their plants to test basically quality control of their products. And that's obviously very widespread in all industries now. Okay, well, now here we are, it's 2026. Everybody talking about big data, AI, all that kind of stuff. It's like, well, maybe we don't need experiments anymore because we have such a huge amount of data coming in from the real world that we don't need these small scale experiments on 24 mice or whatever anymore. Well, I think it's still a complementary approach that can still be important, because it's still the standard for learning about what causes things in the world. No matter how much big data you have, you still don't have that control unless you do an experiment. Okay, so that was the history of experiments, at least a very, very short version. So I'm going to, again, please feel free to ask questions and interrupt. we'll talk about the concepts of experimental design. And these are things that you all need to be aware of in the experiments that you're working on. Whether you are the one who designed the experiment or not, it's important to understand how the experiment was set up so you'll know how to analyze the data that come out of it. So here are some of the terms that we're going to be talking about. I'll mention them individually as we go. So first of all, treatment. What is a treatment? You'll see that term in the literature, right? Because you know, experiments, randomized controlled trials use the term treatment because you're treating a disease, but we use the term treatment in all experiments, even if it's not a medical study. It's anything that we manipulate, any condition that we impose on the subjects of an experiment. And a factor is any variable whose levels are set by the experimenter. So like if you have a drug, the factor would be whether or not the patient gets the drug, and the levels would be like the drug or placebo, for example. Or you could have more than one level. You could have different dosages. Those would be different levels of that factor. Those would be different treatments. You know, no drug, small dose, medium dose, high dose. Those are treatment levels. And you know, it doesn't have to be doses. You could have in ag science, you could have different management. You know, you could have untreated fields. You could have fields that are, you know, given low nitrogen, high nitrogen, plowing, not plowing, and things like that. Okay, so I use the word control a lot. What is control in the sense of an experiment? What is a control? So a control is any observation within an experiment that shows you what the effect of no treatment is, or a placebo treatment. Placebo being a mock or sham treatment where you go through the motions of giving the treatment, but don't actually have the active ingredient in it. And we use the control to establish a baseline of what the response would be if we didn't treat. And how do we set up controls? Well, it depends what you're controlling for. Because basically anything that you manipulate in an experiment should have a non-manipulated counterpart in the control group. Like if you have 10 people that you want to do an experiment on, you give them all the drug, and they all, you know, get better or whatever. How do you know they wouldn't have all gotten better without the drug? You don't know unless you have that unmanipulated control. And you all might have heard the term positive and negative control before. So again, like I said, there's not just one kind of control. There are different controls depending on what you're controlling for. Negative control is like what I'm saying, the baseline to compare the treatment effect to. A positive control is something that ensures the experimental procedure is working using standard methods. For example, if you're trying to test a novel experimental drug, you might have an unmanipulated control group, a group that gets the new drug, and a group that gets the standard drug that we know is effective. So you can compare and see if that one's working, and compare the results to the experimental new drug, for example. That would be a positive control. And sometimes it's not possible or it's not ethical to have an unmanipulated control. For example, if you have human subjects and some of them have a bad disease, it would be unethical to not treat the disease at all. So we have to work around that. And in some cases, you know, the best, second best thing is to get baseline measurements on the subjects before you do the treatment. So you have kind of a pre-treatment baseline on those same individuals. Then you do the treatment and compare. So that's also a type of control. It's not as good as having an unmanipulated group, but in some cases it would not be ethical to have untreated subjects. Okay, this is something that Matt and I were talking about yesterday, and so it does come up in your stuff. What is an experimental unit? An experimental unit is anything or person or animal or object that you independently, randomly assign the treatment to. So like if you're doing an agricultural study, you have a big field, you divide the field up into areas and you randomly assign a particular area to be planted with one variety of crop and another. So it's random which piece of land gets which crop. So that little plot of land is an experimental unit. Or like if you're trying to test a vaccine and you have a bunch of animals, you randomly assign a mouse to get the vaccine or not. So every mouse has an equal chance, has the same probability of being in the control group or the group that gets the vaccine. So the mouse is the experimental unit in that case. It could even be in the Forest Service, they do studies on entire watersheds. So like a huge piece of forest is an experimental unit and they assign them treatments like this one's going to get logged, this one is only going to get some of the trees cut down and so forth. And so the experimental unit, this is an important point to make. The experimental unit is not necessarily the same as the observational unit that we make measurements on. So we were talking about this in the context of single cell omics, right? Like from each subject, you might get thousands or more of cells. And you have omics data on each of the cells. Well, you don't really have thousands of sample size now because those cells are not experimental units. They were not independently randomly assigned a treatment. They all come, like those 1,000 cells all come from the same mouse or the same person who is your experimental unit. Those cells are only observational units. So if you did your statistics, assuming that each of those cells was independent sample, you're going to get probably wrong results or an inflated idea of how much of an effect your treatment has if you don't properly account for that those cells are not independent units. So that's just an important thing to keep in mind. But I mean, you see it in the literature all the time that people are doing this wrong and getting wrong results. So, just because something's been published in a journal doesn't mean it's right. In fact, it's usually wrong, to be honest. So yeah, I may skip over some material later, but I think some of this stuff's important to talk about how experiments are set up. Experimental design has three main components. The treatment structure, those are what are the treatments and which unit gets which treatment. We have the design structure, how treatments are assigned to experimental units and how the units are structured in space and time. Like if you have in your mice studies, you might have cages and multiple mice are in each cage, the physical layout of the cages. If you did multiple runs of the experiment, on different days or times, that would also be part of the design structure. And then the response structure, what responses will you measure and how does that relate to the experimental units? That would like be if you have a mouse and you measure some response on it every day for 10 days, then you have repeated measures in time, even though that one mouse is just one experimental unit. Or like the single cell thing, you could have many measurements, many cells from the same mouse. That's all part of the response structure. And this also applies even if you don't have a controlled experiment, all of these things still apply to an observational study. Like if you have just patients that you're collecting data on different days and about their except for the treatment part, the other points still apply. So yeah, treatment structure, we have a lot of different designs. You may have heard these terms, and some of your studies are set up along these lines. Like you have a one-way design, like if you just have one factor, like drug versus no drug, or doses of one drug, that's a one-way. Factorial would be like if you manipulate multiple different factors and do all their combinations. Like if you have, you all have studies where you have, some mice are on standard diet, some are on a high-fat diet, and then within each diet, you do some intervention like exercise or something like that. So you're crossing diet and exercise. That's a factorial design. There are also designs where you're manipulating a continuous variable at different levels, like that's a regression design, and they get more complicated from there. Like I said, factorial would be one with multiple factors you're manipulating. Yeah, that's more of a, I don't know if you all do this type of studies that much. They're very common in agricultural studies, but also in medical studies. Genotype by environment interaction is very important to look at. Like, for example, if you have, if you want to know, like, does smoking cause cancer? Well, there also might be some genetic effect there. Some people might be more likely to develop cancer based on their genetics if they would smoke. But if you're not put in the environment, like if you never smoke, then you would never see that genetic effect. So you have to look at the genotype by environment interaction, and you can do that with an experiment. Obviously, it's not ethical to do experiments on humans with tobacco smoke, but you can do it with other things. Now, the more different factors you want to manipulate in an experiment, the more realistic, right? Because it's not just one factor that changes in the real world, it's many things. So if you have many different interacting factors, you can kind of get closer to realism, but the more you're going to need tons of combinations, right? If you have 2 factors, each with 2 levels, you'll need 4 combinations. If you have 3, you'll need 8 combinations, 16, 32, and so forth. So and if you need multiple samples in each of those groups, you're going to need a huge sample size if you have a lot of interactions. Not to mention, sometimes it's very hard to interpret multi-way interactions statistically. So there's kind of a trade-off again between the realism of having many interactions and the control of just having one factor you're manipulating. Let's talk about the principles of experimental design as were first put together by R.A. Fisher. So these are the 3 things you have to take into account when you're designing an experiment. Randomization is the first one. So it must be random which units get which treatment. So like when R.A. Fisher first did his first agricultural experiments, he took a deck of cards, shuffled them, and then based on whether you got clubs, spades, diamonds, or hearts, decided which treatment each plot would get. People were like, well, a deck of cards, like, what are you doing? Like, that's not very scientific, but it's, but as it turns out, if you don't have each unit have an equal probability of being assigned to each treatment, you may overestimate or underestimate the effects of treatment. So it should be random. Replication, we need to not just give one mouse a drug and one mouse placebo. We need to have many within each treatment group because we want to estimate the variation within treatment versus between treatments. That's like the basic idea of an ANOVA would be to look at the variance within and between. And we can't know what the variation is within a treatment unless we have multiple units within each treatment. Replication. And local control. That means the conditions should be as consistent as possible across treatment groups. So like if we, this is not as relevant in highly controlled lab studies like you all are doing, because the idea of having a controlled lab environment is so that the conditions are very constant. But if we're doing experiments in the field, you definitely need to take that into account. Like you have to have, you don't want to do an experiment on a field that has very different soil types and put one treatment in one soil type and one in the other, because that's going to confound your treatment. So you want to make them as consistent as possible. And we'll talk about in just a minute ways you can do that. And again, if an experiment is well designed, if you have good lab protocols, you can minimize the influence of any variables besides the ones you're testing. We do have statistical methods to account for them after the fact, but statistics aren't as good as having a good protocol in your design. It's better to have a well-designed experiment than to try to sort of bail yourself out by using statistical models to deal with things that you should have taken into account in the design process. Yeah, so we do have stats, but still, that's no reason not to have good protocols and do things cleanly. So there's this concept called optimal design that I'll talk about. Basically, if you have a statistical model, you're trying to estimate something like the effect of a drug or whatever, then there's sort of a best design that will give you the most efficiently be able to estimate that outcome that you care about. Like if you have a certain number of mice or agricultural plots of land or whatever, and you have a scientific question, there's a design that maximizes the statistical power to answer that question. Power being the probability of detecting a phenomenon that exists. And some people call that efficient design. So, there's background noise, variation that we don't care about, and then there's variation due to the thing we care about, which is the signal. We want to maximize the signal to noise ratio so we can estimate the thing we care about. And there's two things you have to account for when you're estimating, which is bias and variance. Bias is how far our estimate is from the truth, and variance is how uncertain the estimate is. So we want to minimize both those things. And as I said, there's not just one optimal design for everything. It depends on what we're testing. So I will go over a few different, this part is maybe not as relevant to you all if you're doing a lot of lab studies. So I'll kind of just gloss over this, but it is very important for people doing field experiments. The simplest experimental design is a completely randomized design, or CRD. And there's no grouping of experimental units. Every single experimental unit is independently randomized. Like if you had a mouse experiment and had mice that were housed individually in cages, each cage has only one mouse, one experimental unit within it, and there's no grouping of experimental units. Each mouse is completely random which one they're assigned to. Like if you had 3 genotypes and 2 treatments, a 3x2 design, there's 6 combinations, and you have, in this diagram, there's 4 replicates per combination, but it's completely random how they're positioned physically. They're all completely mixed together. So there's no local control there. We're not trying to have local control. We're just completely randomizing. And, in a highly controlled lab environment, this is probably a good, if not the best design, but it is not the most efficient for field environments where if you have physical variation in the environment over space, then you want to sort of space your treatments out so that every treatment combination is present in all the physical environment variation, so you can have local control for that environment. And the way now you still can account for the environmental variation even with a completely randomized design, because there are statistical models that you can use to account for it. But again, it's better to have good design than to try to bail yourself out later with statistics, I think. So blocks are what people use for local control instead of using a completely randomized design. A block is a group of experimental units that are somehow related to each other. Like if in your mouse study, if you have cages and you put multiple mice into one cage and like, okay, this cage has mice that are given placebo and this one has mice that were given a drug, but each cage might have 5 mice in it or whatever. Those mice are still independent experimental units because the individual mouse was randomly assigned to a treatment, but they are grouped together with other experimental units. So to be in a block together, that means those units are close together in space or time. Like if you did a study on mice and did 3 repetitions of the trial on different days, each day would be a block as well, because it's possible that the lab conditions could have varied from day to day. So those units that were on day 1 are more related to each other than units from different days. Space or time can be a block. And so blocking is good because it allows us to estimate what the effect of other influences in the environment are besides the one we're manipulating, and then we can get a better estimate of the actual effect of the drug or the treatment or whatever it is. And so you can actually get more statistical power from a block design than you can from a completely randomized design, depending on how the environmental variation shakes out. Yeah, so you could think of it in an agricultural field. You divide up the fields into groups of plots. Each of those group of plots is a block. Or like if you're doing a study on school children, like if you wanted to test some new educational intervention, a new method of teaching math or something like that. Well, you don't just want to do your study at one school because there could be something unique about that school. You want to do it at many schools to see if the method works well in general. Well, you sort of have to, you can't just give one student at a time the new method. You have to, tell the teacher of a particular classroom to use the old method or the new method. So, each classroom is the experimental unit because the classroom was assigned the intervention, and then each school would be a block. Like, a high resource school in a rich neighborhood might have different effects than one that's in a poor neighborhood or whatever. And and so then, right, the classroom is the experimental unit, but the individual students are going to be the observational units because you might give each one a test or an assessment, but those students from the same classroom are not independent or from the same school as well, if that makes sense. And obviously if you're doing a study of some medical treatment in humans, you'd also want to do it at multiple hospitals if possible for the same reason. And a hospital would be like a block containing multiple experimental units, which are the patients, to which is randomly assigned to a treatment. So yeah, that would be where the RCBD, randomized complete block design, comes in. Basically, this is the simplest form of block design. Each block is a complete replication containing every treatment combination once. Like if you had, like the school example, you might have, at school A, we're going to give one classroom the old method and one classroom the new method, and do the same at school B. Each school has a complete replication of all the treatments that are in the experiment. It could have multiple replications, but the simplest RCBD is each block contains all the combinations. Then, if you're in the ag world, you have a split plot design where you basically have, if you have more than 2 treatments, then you randomize one at a larger level and one at a smaller level. That can actually have more statistical power for certain questions than like, especially if you're trying to look at an interaction between 2 treatments, it can have more statistical power than a randomized complete block design. I'm trying to think of, there are examples in clinical trials that would be equivalent to a split plot. I'm trying to think of an example. It would be like if you had, if you had like some sort of intervention that you were doing at the level of an entire hospital, like you changed something about that environment, and then within that, you would have patients that were individually assigned to different treatments. So like one treatment's applied at the level of the hospital, the other is applied at the level of the patient. That would be a split plot design. And it can also be used for logistical reasons. Like if you in an agricultural study, you might have an irrigation treatment, but you can't just irrigate a tiny little patch of land. You have to have irrigation on the entire field. So you'll have a whole field that's irrigated and one that's not. And then within that field, you have different crop varieties planted. That's also a split plot design. And yeah, it has more power to detect effects of the treatment that's randomized at the smaller level and the interaction effect. So if those are the ones you care about, it's actually better to do it this way than to do a randomized complete block. Now, this applies, I sort of said this for the for the ag scientists, the crop scientists in the Ag Research Service, they're like obsessed with randomized complete block design. Every time they set up study design, they only ever do randomized complete block. They're not even aware of the other options that are out there that may actually be better, and you can get more bang for your buck with the same amount of money and time spent. You can get better statistical results, more power to get the answer you care about if you use the right design. So I won't go into the specifics of it, but basically you should talk to a statistician before you start collecting data, because you might be able to actually, people always complain like, well, I always tell them they have to have more replicates and things like that. It's like, well, it's easy for me to say that. I'm the one who's like there on the lab bench, like doing all the replicates, or out in the field, like breaking my back, doing whatever. But sometimes I can actually help you and give you better results for the same or even less amount of effort. So hence, if you just do a little work to get the design right, then you can actually save yourself effort. So yeah, I'll just kind of gloss over this because it's not as relevant to what you all are doing. There's sort of incomplete block designs that can have more statistical power if you have like hundreds of genotypes you're comparing in a trial. I'll just kind of gloss over that since you all probably won't work on that stuff. And augmented designs for the same reason, that's more relevant to ag scientists. Yeah, and there's strip plot, there's Latin square, like a Sudoku board, basically. There's all kinds of designs that can be more efficient for particular questions. So I'll skip that. Oh yeah, this is somewhat relevant to you all. I mentioned this already, that blocking or local control can be done in space, but also in time. All of the things, the diagrams I showed show a spatial arrangement, but actually time can also be a dimension that you can use for blocking. Like if you did different runs or different batches, those are considered blocks. Because I know some of the data I've helped you all with are like you did behavioral trials, but that are done on different days and that kind of thing. So those would be blocks. And if you have, some sort of environmental chamber, like a lot of scientists I work with have these environmental chambers that are very expensive, and you can perfectly control the conditions, but they only have like 2 or 4 chambers maybe. So you don't have very much replication if you just do one run of the study, since the entire chamber is assigned to a treatment. So the only way you can get replication is by doing the replication in time and do the entire study on those 4 chambers and then repeat re-randomizing which chamber gets which treatment, but then each run in time is a block and you account for that statistically. So yeah, there's the idea that you can replicate experiments in space and time. Again, this is more relevant to people that are doing field experiments. If you do an experiment in multiple locations in multiple years, or not necessarily years, but multiple times, you want to re-randomize every time. Like if you have mouse cages, every time you repeat the experiment, you want to reassign, re-randomize which cage gets which treatment, because that ensures that within each trial, every experimental unit has an equal chance of being assigned to each treatment. Now, there's also the concept of treatment by block interaction. So all the designs I showed you, the randomized complete block design, allow you to account for differences between the means of blocks, but what if the relative differences between the treatments vary by environment? Like with the school example, right? Like if you're trying to test a new educational method, maybe certain students from particular socioeconomic backgrounds, the new intervention is better for them and has a greater difference from the old method than others. Well, you can't really estimate that interaction between treatment and block unless you have replication of the treatments within blocks. So it's good to replicate one or more treatments within each block. Like with the school example, you could have within each school, 4 classrooms that got the old method and 4 that got the new. Then you have a treatment. You can estimate that treatment by block interaction. So, sorry, does anyone have any questions before I do the last few slides here? I should have said, yes, Peter.
So like, let's say like you brought up the example of like, you have like a field and you're like testing like treatment and like the field's like uneven and
Right.
it's a case where you should have blocked, but then like you didn't. Like you mentioned, like there's still ways to account for that statistically. Like how would you go about that?
So yeah, basically you could, even if you didn't block, you could assign a spatial, you could basically include a spatial term in your model that would account for the variation over space statistically. But I'll mention this in the mistakes section in a second, but if you didn't randomize properly so that like all of the plots from one treatment were in like the north side of the field and all which has, let's say more prone to flooding, and then the other treatment is on the south side, you're kind of stuck because you have confounding variation there. So like, if you didn't set up the experimental design right, like no statistical method can get you out of that problem. Because if you see a difference, you're never going to know for sure if it was because of the treatment or because of the environmental variation. But yeah, but the spatial term can get you out of that problem. So yeah, basically this leads into the next section, which is like, well, the mistakes that we make in experimental design are if you have too much variation within your blocks, if you do pseudo replication, or if you don't have randomization within blocks. So yeah, I see this a lot in agricultural studies where like people will have these really long skinny blocks that have like 100 different genotypes and all in one row. And it's like, well, really blocks are supposed to represent consistent environmental conditions within each one. If you have a long skinny one, the two ends of that block are really far apart. So they're not really, the one at the far left and far right is further from each other than they are from ones in a different block. So those aren't really good blocks. It's better to have like more compact ones that are more square than long and skinny. Pseudo replication, this is something again that comes up in the single cell world, where that means that's a term for when you incorrectly consider experimental units to be independent when they're actually correlated, or if you consider a observational unit to be an experimental unit when it shouldn't be. So that's what pseudo replication is. Again, that would be like calling all the 10,000 cells from one individual to be independent units. And it can lead to confounding where we can't distinguish between different influences on the outcome. So like, that particular mouse that you got the 10,000 cells from might just have higher gene expression for a particular gene just because it's that one individual's like quirk. But if you consider those 10,000 cells to be independent, you're like, wow, the treatment had a really big effect on the gene expression of that gene, but really it might not be. It's just that individual mouse had a high expression. So yeah, sometimes it can be okay, but you have to just consider what population you're making inference on. So like I saw this with somebody who was doing a study on chickens, and it's like, well, we want to know if free range versus caged hens have different egg quality. Well, they just had one facility or one barn that had all free range and one that had all caged. Each one maybe had hundreds of chickens, but you don't know if it's really because of just specific differences between that specific barn or whether it was because of the free range effect in general or not. So like really, when you do that experiment, you're estimating the difference between those specific barns and not between free range and cage overall. So you're actually making a conclusion about a much smaller population than you think you are. So yeah, this is like what Peter was saying. This experimental design, which is supposed to be a randomized complete block, is actually a poor randomization It would be fine if you replicated that whole thing multiple times, but if this is the whole experiment, it's like all your treatment 1 is on the left or on the west side, all your treatment 4 is on the east side. So if there's an environmental factor that varies from east to west, it's going to be, you're not going to be able to tell if it's the treatment effect or the environmental effect. So yeah, your spatial term will help a little bit, but there's always going to be some confounding variation there unless you randomize within each block. You know, you would want to shuffle around the 1, 2, 3, 4 within each of the rows. And that's how you would make sure that each treatment occurs equally often within each environment. So, yeah, what kind of statistics do you have to use for these design experiments? And this is relevant to you all since these are the statistical models that we use, or that I, in collaboration with you, use to analyze your data. I mentioned statistical power already, which is the probability that if there is a true effect in nature that exists, what is the probability that our statistical model will be able to identify that effect? Like if you're using a p-value, the probability that we'll get a significant effect if that effect is real. And it depends on your sample size, right? If you have more replication, you have more probability to detect the effect because you have more precision on your estimate. It depends on your experimental design, like I said, and it also depends on what effect you're trying to estimate. If you're trying to estimate a very weak effect, you have low power unless you have a very big sample size. Versus a strong and obvious effect, you wouldn't need as much sample size to get the same probability of detecting that effect. And usually, so you all may know about this from, if you do experiments with vertebrates like mice, you have to get your experimental designs approved by a review board before you do the experiment, because it's considered unethical to waste, to kill animals for an experiment that is poorly designed. And so statistical power comes into that. You're required by IACUC to write down what your experimental design is and to estimate the statistical power based on your design. And right, because they don't want you to use an excessive number of mice, but also if you use too few mice, you're not going to be able to draw any conclusions about your data. So those mice also were sort of wasted in a sense. So you want to hit the sweet spot and get the sort of minimum number that you need to get the appropriate amount of statistical power, right? Because power is kind of a balance between false positives and false negatives. If you have too few sample size and you don't find a significant effect, maybe there was no effect, or maybe you just didn't have enough power to detect that effect that really was there. So that would be like a false negative. Yeah, so basically, anytime we do statistics on data that come out of an experiment, we want our statistical model to represent every possible thing that we think was influencing the result. That's what we call the data generating process. And that includes the experimental design. And a tool that we as statisticians use to analyze data from design experiments is the mixed model. A mixed model is a statistical model that includes fixed effects, and those are effects that are consistent from experiment to experiment. Like if we are doing an experiment to test the effect of a drug, that the drug term in the statistical model is a fixed effect, because if somebody else wanted to repeat that experiment another time, or if we wanted to repeat it again, we would use those same levels in that experiment. But a random effect is one that is unique to that particular study, and we model them as being drawn from a random population. Like if we, again, like with the school example, if I did a study on schools in North Carolina in 2026, testing this method to improve math skills, well, if somebody else wanted to repeat it in Arkansas in 2027, they would use different schools. They have no choice. And so the effect of the individual schools, we are modeling those as random effects. They're sort of drawn from a random population of schools, and they're unique to that particular experiment. And the sort of math of how the effects are estimated between fixed and random is different. And so that's why we use mixed models for experiments that are designed. So whether something is a fixed or random effect, we usually consider our treatments, like I was mentioning, the drug or the educational intervention or whatever is a fixed effect, and the blocks are random effect, the cage that multiple mice are in, or the school that multiple classrooms are in, and so forth. Right, like I said, if someone repeated your experiment, they'd use the same treatments. I just said that already. So yeah, but sometimes it can depend on your question whether you treat an effect as fixed or random. Like if you did repeated measures of body weight or of metabolic rate on the same mouse, you might like, is time considered a fixed effect or a random effect in that sense? Like if you did it at different time points, it depends on your question. If you're actually trying to estimate the effect of time, then yes, you would treat it as a fixed effect. But if you are just trying to measure that mouse multiple times to make sure you have a precise estimate of the metabolic rate, and you just have to get multiple measurements to make sure you estimate it right, then it would sort of be then the repeated measures within the same mouse would be a random effect. Yeah, because, you know, I put the thing from Harry Potter, because you can never go back in time. You have to, you know, if you repeat an experiment over time, you could never exactly replicate the environmental conditions from a specific time or the lab conditions or whatever again in the future. It's completely unique. So yeah, you know, it depends on your question. But to me, the fixed versus random thing is not the most important thing. The most important thing is to make sure that somehow your statistical model includes all the things you think are relevant or that might affect the thing you care about, so that you can more precisely estimate the thing you do care about. And yet, if you're not sure, please talk to me or another statistician. I know you have other statistical folks here that you can consult with. Yeah, so just concluding a few slides here. There are various resources available depending on what statistical software you use, different packages and stuff like that. I don't think you all use SAS that much, so mostly there's a lot of good R packages, and then there's this like page called a CRAN task view that you can click on the link in the slide that shows you all the R packages that are related to experimental design. So to just give a couple slides to conclude, I want to just since I've been talking about how great experiments are, here's kind of the opposite side of the coin, just so you don't get too married to the idea of experiments. So I mentioned R.A. Fisher and was talking about how great he was for inventing experimental design and ANOVA and things, but Fisher had his kind of dark side as well. So like there he is smoking his trusty tobacco pipe. That's a page from National Enquirer, not a very reputable newspaper. Basically, we've talked about how, okay, an experiment is the best way to get information about, does X cause Y? If we just observe nature, we don't know what caused that. We have to control with an experiment. But he kind of took that idea a little bit too far, or I would say even weaponized that idea. He was paid off later in his career by the tobacco industry to advance this idea. It's like, well, you know, it wasn't really known for sure that tobacco use increased the risk of cancer. That was like a new idea at the time. And obviously the cigarette manufacturers were trying to kind of suppress that idea. So they hired R.A. Fisher to go out here and discredit anybody who was trying to establish this link by saying, well, we can't do a randomized controlled trial on smoking versus non-smoking. So just because cancer rate is higher in smokers than non-smokers doesn't mean that we know that smoking causes cancer. Like, well, maybe people that already have lung pain because they're like about to get cancer smoke to relieve their lung pain. So that's why smokers have more cancer. You know, those were things that he said. And you can see that it said cigarette cancer link is bunk, was what he helped put that out there. So, you know, basically, yes, it is true that a randomized trial, a randomized experiment is the best or one of the best ways to conclude something about causality. Doesn't mean it's the only way or the best way to get knowledge. And as Peter and I were talking about yesterday, there's like these causal inference methods that we can now use, statistical methods to infer causality from observational data without doing a controlled experiment. So like if you can take observational data, and if you account for different confounding factors and variables, you can actually estimate the causal effect of smoking on cancer, even without doing an experiment, as long as you have the right data and the right other variables that you measure. Yeah, so, and another thing I mentioned, okay, yes, we can almost establish or even, you know, have a lot of evidence in the lab in controlled conditions that X causes Y, but a lab experiment is artificial, right? It does not, even if your experiment is perfect and statistically 100% robust, in, you know, then that does not prove that something's going to work in the real world. You know, maybe the drug only works in the lab for whatever reason, and when people are exposed to natural conditions in the environment, you know, not just mice in a lab, it doesn't work. So you have to go out and get real world data as well as controlled experiments. So my final recommendations are, when you're designing an experiment, think about all the different components, how the treatments are assigned, and when you're measuring and what you're measuring it on. It's really important to know and be explicit about what question you're trying to answer. And to just be aware what you're controlling for, and to strike that balance between control of other variables and realism. And, you know, it's good to have a lot of interactions and manipulate different factors. You know, that's more realistic, but if you overdo it and try to stick too many factors into one experiment, which I see all the time, people set up these really complicated designs, but then don't even know how to interpret the results they get. So it's like, well, why didn't you just do a simpler experiment? And then you could have interpreted and actually, you know, been able to understand the results you got. And yeah, again, like I said, a simple design is sometimes good, but there might be other designs thinking outside the box that will give you better results for the same amount of replication. So why not be creative? And you all sort of have to do this if you're working with mice or humans, but it's always good to, if you can, do a power analysis, estimate what the statistical power of your design is going to be before you do the experiment, so that you know what to expect. And again, yeah, then that helps you. If you do the power analysis, you're sort of forced to think about what your data is going to be and how you're going to analyze it before you even do the experiment, which saves, I mean, I see this a lot, like people give me experiments that are badly designed and ask me to analyze the data. It's like, well, you can't really answer the question you thought you could because it's not the right design. So if you thought about that ahead of time, that saves a lot of trouble later on. So please talk to the statistician before you do the study. Yeah, and that concludes my talk on experimental design. So does anyone have any other questions or comments? Cool. Well, hopefully that gave you some food for thought. So it's about 10:15 now. So I think the plan is, what did I say on the agenda? Do we
Break.
I know we got a break, but when should we start back up again?
And it's chance to show us something to make how to use the power analysis.
Yeah, so I see, I left the gap between, oh, I did that wrong. Well, cool, well, we have plenty of time. So why don't we, yes, we actually, I feel like, why don't we just, you know, take 20 minutes and come back at like 10:40, and that'll give us plenty of time to do what we need to do, for the remainder. So yeah, why don't we just come back at 10:40? And I'll be around to like chat about whatever in between. Stop the recording for now, so we don't have a lot of not recording. Sure.
I